This narrated presentation discusses evidence for a genetic contribution to the etiology of idiopathic normal pressure hydrocephalus. I'm Rita Dossi from the University of Massachusetts.
And I'm Mark Johnson.
Idiopathic normal pressure hydrocephalus, or INPH, is a neurological disorder of aging that is characterized by ventriculomegaly, gait apraxia, urinary incontinence, and dementia. It is estimated that approximately 700,000 Americans are affected by INPH, with the average age of diagnosis being 75 years. Risk factors for INPH include hypertension, hypercholesterolemia, diabetes, and alcohol consumption. The physiological mechanisms underlying these associations are not known. Abnormal signs observed in INPH patients include enlarged ventricles, increased amplitude of intracranial pressure waves, ventricular CSF stasis, and cerebrovascular changes.
Radiographically, patients with INPH are noted to have enlarged cerebral ventricles.
Gait dysfunction is the most commonly observed symptom of INPH, occurring in about 90% of cases. The gait dysfunction of INPH is characterized by decreased step height, decreased stride length, decreased cadence, and impaired balance. The gait can be described as magnetic, bradykinetic, shuffling, or short-stepped. The gait dysfunction appears to be the manifestation of apraxia. Weakness is not commonly observed.
Unlike common neurodegenerative disorders such as Alzheimer's disease or Parkinson's disease, the symptoms of shunt-responsive INPH can be improved by CSF drainage. Indeed, ventricular enlargement, the presence of the characteristic INPH symptoms, and a response to shunt placement are necessary for establishing a certain diagnosis of shunt-responsive INPH. This graphic describes the path to diagnosis of shunt-responsive INPH.
Numerous reports of familial INPH have been published in the literature, including several pedigrees with autosomal dominant transmission. Table 1 shows the familial reports of INPH in the literature. One of the earliest reports of familial INPH was made by Portnoy et al., 1984, who described shunt-responsive INPH in a 67-year-old man and his 74-year-old sister. Zhang et al. Reported a familial disorder which they called essential tremor idiopathic normal pressure hydrocephalus, or ETINPH. They described a family spanning 5 generations that first developed essential tremor, and later, after the age of 65, family members developed INPH in an autosomal dominant fashion. In 2011, Cusimano et al. Reported a case of 2 sisters who developed shunt-responsive INPH in their 7th decade of life. In the same year, Takahashi et al. Reported a pattern of autosomal dominant inheritance of INPH in 4 patients spread across 3 generations. In 2014, a similar report of 2 sisters after the age of 70 with INPH was identified by Luita et al. Interestingly, both of these patients had daughters who developed urinary incontinence, and findings on MRI consistent with INPH after the age of 40. Magir and Cusimano reported a well-characterized case of familial INPH and conducted a case control study of the occurrence of the INPH symptom triad among relatives of 20 INPH patients and relatives of matched controls. In this study, just over 7% of INPH patients had multiple first-degree relatives who exhibited at least 2 or 3 symptoms of the INPH triad. In 2016, another team used multivariate logistic regression analysis to analyze a nationwide cohort of 375 shunt-responsive INPH patients and their families. Results from 12 separate pedigrees revealed that 4.8% of patients had at least one INPH relative who required CSF shunt placement. More recently, Morimoto et al. In 2019 performed whole exome sequencing of DNA obtained from a Japanese family affected by INPH. There were 2 affected individuals and 3 unaffected individuals. They identified a heterozygous truncating mutation in the CFAP43 gene in both of the affected individuals. A study in 2017 examined 23 candidate Alzheimer-associated single nucleotide polymorphisms in 188 shunt-operated INPH patients and 688 controls without diagnosed neurodegenerative disease. Of the 23 candidate SNPs, logistic regression analysis identified allelic variation of NME8 between INPH patients and controls. Mutations of NME8, also known as TXNDC3, cause primary ciliary dyskinesia. In 2018, multiple groups became interested in the gene SFMBT1. A group in 2018 found that 10% of Finnish and 21% of Norwegian shunt-responsive INPH patients had copy number loss in the second intron of the SFMBT1 gene, whereas only 5.4% of healthy Finnish elderly individuals had this genetic change. Morimoto et al. In 2019 performed whole exome sequencing of DNA obtained from a Japanese family affected by INPH. This was referred to in the last slide. They identified a heterozygous truncating mutation in the CFAP43 gene in both of these affected individuals. CFAP43 had previously been shown to play a role in the function of sperm flagella, and mutations in the CFAP43 gene cause male infertility. Morimoto et al. Created a CFAP43 knockout mouse using CRISPR technology. Mice homozygous for deletion of CFAP43 developed hydrocephalus and infertility.
This is a video of live ciliated ependymal cells from the mouse brain ventricle showing fluid moved by the actively beating cilia.
This graphic was taken from the study by Morimoto et al. In 2019. Here we see characteristically enlarged ventricles in the affected individuals, on the left. This graphic from the paper described in the last slide reveals the ciliary ultrastructure of the nasal mucosa in an effective INPH patient. The blue box reveals normal 9+2 axoneme. The orange box reveals abnormal compound cilia involving 2 axonemes in one ciliary shaft. This graphic is from a paper in 2007 which revealed that mutations in TXNDC3, also known as NME8, cause abnormal ciliary structure with absent dynein arms in affected individuals, labeled here as D50X1. As you may recall, allelic variation for the gene NME8 has been noted between INPH patients and controls.
To date, surveys of INPH patients have identified a possible affected family member in 5 to 10% of cases. The finding that genetic variations involving CFAP43 and NME8 are associated with INPH raises the possibility that some cases of INPH may be caused by defects in ventricular ciliated epithelia. Intron loss in the SFMBT1 gene, which has also been associated with INPH, occurs in 10 to 21% of Finnish and Norwegian INPH patients. This finding suggests that at least 10 to 20% of INPH patients are genetically susceptible to developing this disorder. This number represents a minimum estimate, since it focuses only on SFMBT1 and does not consider mutations in CFAP43, NME8, or other yet-to-be-discovered INPH-associated genes. Additional genetic studies of patients with sporadic INPH are needed to more clearly establish a causal role for genetic alterations in the etiology of INPH.